Okay, let's have a class. This is last lecture before the break. So next week, Tuesday, we don't have a lecture. We have a lab midterm exam. The first lab will be start at 11 o'clock. Then the other lab sections will just based on your lab sections. The exam room will be in the lab room. And next week, Thursday, we don't have a class because it's a fall break. It's been a long time. The Thursday has been included into the fall break. Usually it's only Friday, so we cut one lecture. We will be postponed the exam 2 date. Will be one date after based on the syllabus. Okay, I think the syllabus says on Tuesday we're going to move on to Thursday. Either way, we will be moved one day later. Okay. We're going to talk about the human disease caused by bacteria. Basically focus on Clostridium today, but before that, I will do the review what we already talked. So we're going to using a different way to do the review. I think I'll just draw in here in the middle. So what we talk about is Streptococcus. And then we also talk about the Staphylococcus. So this is the one which is a coccyl. So we find that they are gram-positive coccyl. This is also gram-positive coccyl. Gram reaction morphology are the same. Arrangement. Ideally, this is chain shape. Or we say Diprococcyl, very short chain, only 2. And the Staphylococcus aureus is like this. We say grape shape. However, you should know, this is a very theoretical model. In the real life, it's very difficult to differentiate. So think about it. You are working in the hospital. And you get a sample, a sputum sample from a patient who had a pneumonia. And then they say, I'm going to do a gram stain from a selected media, and then I'm going to be perfectly to see, oh, this is gram-positive coccyl chain, that's Streptococcus. The other one is Staphylococcus aureus or epidermis. It is pretty much difficult. So we have to rely on other things. In the microbiology clinical area, the other test also has to be quick. So you cannot be doing like sequencing or took like 5 days to get the results. So we have to rely on the biochemistry test. And there is a very quick biochemistry test. Could determine either one of that. Which means if you end up your pathogen, one way or the other, it's either Streptococcus or Staphylococcus, but I just don't know. I have to determine. There is a quick way to do it. It's called the catalase test. And the catalase test we will do in the lab. Streptococcus is negative, Staphylococcus is positive. It's a very easy way to do it. How to do it? We're using hydrogen peroxide. If bacteria has catalase, they will have water and oxygen generate. What the oxygen means? Which means it will be bubbling. So if you see on the agar plates, it has bubbling happens, it means catalase test positive. Staphylococcus aureus positive, Streptococcus is negative. A very easy way to determine. But there is one tricky thing. The catalase test can never be done on the blood agar. The reason is blood agar already have oxygen there. You will get false positive. We'll talk about that again in the lab when we move to that. But that's the quickest way to do. So what we talked about, we could do some review. Like we did it before. Streptococcus based on blood agar. We have a dark green colony showed we call alpha hemolytic. And we also could have a transparent zone comes out, and this is we call beta hemolytic. Now, alpha hemolytic, what's the streptococcus we have? That is Streptococcus pneumoniae. And a viridam streptococcus. Let's say Streptococcus mitis, bovis, mutans, all those. Now, how we differentiate those two? We rely on a test. Remember, we call a P-disc. What's the components of the P-disc? Optichan. Streptococcus pneumoniae is susceptible. And the Streptococcus, a viridam streptococcus, is surrounded, the P-disc they still grow, we call it resistant. Beta hemolytic, we talk about the two pathogens. That is Streptococcus pyogenes and Streptococcus agalactiae. So how we differentiate that? Again, we're using a blood agar, but this time we're using A-disc. And what's the components of the A-disc is basic tracing. So Streptococcus pyogenes, susceptible. Transparent zones around the disc. Streptococcus agalactiae, it is resistant. And among all of these, we have a separate one comes out, which is gamma hemolytic. That is Enterococcus faecium or Enterococcus faecius. So this is a big picture right here. Then you can go ahead to talk about it. What is a pneumonia we talk about? We talk about this is 80% CAP, community acquired pneumonia. What's a viridam streptococcus cause? Endocarditis. So remember we said 75 years old grandma took out of the wisdom teeth, has a contamination. What is a Streptococcus pyogenes? That's strep throat. And we also talk about, this is a very invasive one. We have impetigo at the beginning, then cellulitis, then we have a strep throat. How about Streptococcus agalactiae? This is, we say, woman at 35, 37 week of pregnancy need to do a screening test. And this guy we also call Rebecca Lansfield group A, and this is Rebecca Lansfield group B. So we call it GAS and GBS. And the Enterococcus faecium, we call it GDS, group D streptococcus. Now, Enterococcus faecium, we talk about, this is resistant to 6.5% salt, bile, salt, and also can grow at bile esculin agar, which is black colony, because esculin reduced to esculitin. So, that is what we talk about the streptococcus. Then, at the end of the class, we talk about the staphylococcus. What we had. We gonna do a different way, okay? We had staphylococcus epidermis. We have staphylococcus saprophyticus. And we have Staphylococcus aureus. And we talk about this is a normal bacteria on the skin surface. This is a honeymoon bacteria caused related to sexual transmitted disease. And the last one we talk about food intoxication. We talk about methicillin-resistant Staphylococcus aureus, we talk about toxic shock syndrome, toxin 1. So this is what we talked about, but what we can differentiate that is based on what we learned in the lab and also in the exam 1, we can categorize them into more. So the first thing we want to talk about is mannitol salt agar. Their growth, the mannitol salt agar will tell us something. So we learned in your lecture, also in your lab, what is mannitol salt agar? This is a salt, can differentiate bacteria whether they're halophile or not halophile. And the mannitol will differentiate that mannitol fermentation, positive or negative. And it's very interesting, the results. First of all, because they are Staphylococcus, so they're all going to grow on the mannitol salt agar, which means they're all going to grow. That's no question about it. But the color is different. Staphylococcus aureus is yellow color. That is mannitol fermentation positive. Staphylococcus epidermis and saprophyticus both are pink original color because that's mannitol fermentation negative. And this is also mannitol fermentation negative. So that's the first thing. We could differentiate that. The second thing, we can use in blood agar. And we mentioned that blood agar is a very good differentiated media based on the alpha, beta, and the gamma hemolytic. So here what are we going to see? Staphylococcus epidermis is gamma hemolytic. Nothing will happen. Staphylococcus saprophyticus is also gamma hemolytic. Nothing will happen. However, Staphylococcus aureus is beta hemolytic, which means you will see transparent zone. That's important. Now, we're going to talk about the other things. Remember we talked about the joke, the honeymoon bacteria. How the doctor can determine the 25-year-old lady will have some sexual activity cause a problem because this is the only Staphylococcus among those 3 resistant to low dose novel biocin. So this is susceptible, that is resistant, and this is also susceptible. That's why we can do a quick test. Another thing we also want to mention, remember Staphylococcus aureus cause all kinds of problems, especially the toxic shock syndrome. The reason is it's very invasive, and one of the reasons we had a coagulase. It can cause clotting of the blood cell. Coagulase test. We also could do Staphylococcus epidermis is negative, Staphylococcus saprophyticus is negative, Staphylococcus aureus is positive. This is called clotting. It is like a glue. It's like the gluing of the bacterial the blood cell. So this is something we can always do as a conclusion. When we learn in the lab and in the lecture how these things can connect with each other together, and it is very interesting. Then in your mind, there is a networking system in your mind. When you solve those case studies, based on these characteristics, it's easy and quick to do the determination from the clinical standpoints. But I have to emphasize that these type of the way is only give you a preliminary and a quick determination. If you really want to know which species it is, or you want to know even serological typing or genotyping, that needs to be relied on PCR and also gene sequencing or those modern biotechnology. Using these traditional methods can only give you is a first impression. But it is important in the clinical area because once we determine it is Staphylococcus aureus, we have to use antibiotics immediately. And sometimes, not only antibiotics, we have to use antitoxins, toxins immediately. So that's why those quick tests, the blood agar, it is important to give you the determination. Okay, so that's why I want to use these two tables, or you can say is a figures to do the conclusion review for what we have been learned. And I hope you go back to read the after-class reading material during the break. At least you read something, then look at those case studies, and come back. We will do some of the review in the classroom, and we can talk about all those details, all those case studies. Okay, so today we move on to the section which is bacteria cause human disease is number 2. We move to the lower body. So we're going to talk about the human digestive tract. This is relatively simple. So you have an oral cavity, you have a mouth, then you have an esophagus, which means you food taking going through there. Then you go to stomach, then pancreas, liver, large intestinal, rectum, finally is anus. Then you have a poop sample comes out. That's very easy to understand. We want to talk about something from the microbiology standpoint. First of all, your stomach area is filled with bacteria. About 100 cells. Why? Because stomach acid. And everybody knows stomach acid is very low. The pH is 1 to 2. But then also very ironic, how come the pathogen still going through there? Well, most of the pathogen has to do acid resistant. So it has to be defending the lower acid in the stomach area first. And obviously most of them they can do. That is also tells us in the food industry, if during the food processing we only rely on the acid, you cannot kill all the bacteria. But obviously in the human body stomach, because of the lower acid environments, very few bacteria are there. Small intestinal, they started to grow. How about the large intestinal, which is a pre-poop sample or human stool sample? It's huge. You have 10 to the power of 12 cells per gram. And most of those are anaerobic bacteria. And the fecal sample, about like 33.3% is bacterial cell. Now, there is another theory we talk about, is why we have our good life. We have a healthy body because our microorganism in our intestinal area, we call it a gut microbial community, is working very well. There's a lot of research saying the gut microbial community is connected with the brain, with the bone, with the liver, with all kinds of the axis, they call it. That's some other things. But they already start to point it out that our microbiology community in our body, especially gut area, is the center of our body. Although there's other people disagree with that, but more and more research showing up that, which means microbiology community in our body is important. So that's something I want to talk to you briefly before we talk about. This slide will explain to you one of the concepts, which is I briefly mentioned, but we want to talk detail here. It's called food infection versus food intoxication. So what means food infection? Food infection, you're eating bacteria. What is food intoxication? You're eating toxin. What means you're eating bacteria? Said very simple. You cook it, they will be dead. So, give you a very easy example. If you have a salmonella in chicken, let's say breast, okay, breast steak, you have E. coli 0157H7 in beef steak. There is an easy way to solve the problem of food infection is you cooking to internal temperature of 71.1 degrees Celsius, roughly 145 degrees Fahrenheit. Because you're killing the bacteria. Why? These are the toxins. Basically, is endotoxin from LPS. It is heat liable. So that's a good example. How about the food intoxication? You're eating toxin. Let's say Staphylococcus aureus. They'll generate exotoxin. And this toxin is heat resistant. When I say heat resistant, is mild heat resistant. Of course, you're using 100 degrees Celsius to boil it, it's going to be killed anyway. It's heat resistant compared to endotoxin. So that means chicken salads, even you cook it. Let's say you do a microwave. The chicken breast, you took it out, you do a microwave, then you take some pieces and mix it with salads again. You still could have had a problem because exotoxins there, if they already been contaminated with Staphylococcus aureus, it will cause a problem. We call it a food intoxication. The heat, mild heating may not solve the problem. We can say that. And here gives you some of the example. Staphylococcus aureus, Clostridium botulinum, and Bacillus cereus. All these three microorganisms is example of food intoxication. Their on-site time is very quick. You can see somehow is 1 to 6 hours. Clostridium botulinum a little bit longer, but it's within 24 hours. And what the symptoms they will have is nausea, vomiting, diarrhea, abdomen pain, all those. It's a very typical food intoxication symptoms. And the on-site time is very quick. Staphylococcus aureus we already mentioned. Clostridium botulinum we're going to mention real quick. And Bacillus cereus is the same as Bacillus subtilis. Is an endospore-forming bacteria coming from the soil. Most likely will contaminate with abusive storage of rice. So let's say you go from Panda Express, you buy a lunch, and you have rice. You eat all the beef, some Beijing beef or what kind of those things, you'll take it out. Then the rest of the rice you put on the dorm bench about a week. Then you could start to see smells, something comes out. And about another week, you will start to see those black dots comes out, and it's could possibly sporization happens, because lots of the rice is originally contaminated with Bacillus cereus or Bacillus species. So that's some of the examples. Okay, let's talk about food intoxication. Regarding food infection, this one is basically a big topic of food microbiology, and Dr. J is teaching that class. And we have a section later on. We're going to mention some of the species, including E. coli 157H7, Salmonella, and Listeria monocytogenes in this section. Food intoxication we will be mentioned here. Okay, next one we're going to spend in time talk about Clostridium. We will talk about these one by one. Now, first of all, we want to give you an overview picture. What is Clostridium? Clostridium spp. Clostridium species. You always remember, this bacteria is very special. It is gram-positive, large shape. It is streak anaerobic bacteria. and endospore forming. Their unique thing, it is a strict anaerobic bacteria. It is endospore forming. Those are the two key things here. Okay, remember the extra points in your exam 1. Why Clostridium species is anaerobic bacteria? Some of you just say because they cannot live without oxygen. And I wording the other way. I said why Clostridium is bacteria cannot live with oxygen. And lots of you just say it's an anaerobic bacteria. That's not the answer. That's just a transphrase. What the question is. So what this means? We learned about that because they do not have SOD. Superoxidase dismutase, and there is no catalyst. So for them, oxygen is a toxin. Okay, and it is also an endospore forming bacteria, and most of these are subterminal location. So this is the first thing we have to know. Second thing, where they come from. They come from soil. And some of those Clostridium is also a natural microbial flora in our body. It's very interesting. It's originally coming from soil. So what are the Clostridium SPP we're going to cover here? Is Clostridium botulinum or botulism, Clostridium tetanus, Clostridium perfringens, and the Clostridium difficile. And there are lots of them we can talk. So we're going to talk about these one by one. I gave you the slides, which is some of the pictures. But lots of them will be in the after class reading and also going to draw on the blackboard. So first of all, Clostridium botulinum or botulism. We're going to talk about is basically 3 types of the botulism. Number 1, we call wound botulism. So where they come from? They come from different types of the wound. Cuts is one of them. Gunshots could be. And automobile accident. All these type of things end up with a wound in your body will have botulism happen. Why? You think about in the tissue. If you have those cuts, surrounding this area is almost microaerophilic to anaerobic environment. So basically, they favor Clostridium botulism to grow. So that's why they will have the botulism comes out. The second one is called infant botulism. So what is infant botulism for? We'll tell you a joke is never feed less than 1-year-old baby honey, because otherwise you have floppy baby. That is what the picture looks like. The reason is botulism, as a human body, you naturally will have some. And also Clostridium botulism could be existing in the honey. Honey is a high sugar component, originally coming from the bees. but also it's going to touch the soil, touch different containers if it's not being well sanitized. And the baby less than 1 year old, obviously, their normal microbial community in the intestinal and the gut area has not been established. So it's very easy to have contaminated with botulism. Now, why we will have a floppy baby? That is weak muscle. All we say is no contraction. So why? This need to go back to talk a little bit about anatomy. So, our muscle contraction, how we do it? We need a chemical. It's called acetylcholine. That is, we call it a motor plate transmitter. Okay, when this acetylcholine have it, it will be activated. The calcium released from SR. What is SR? Sarcoplasma reticulum. If you learn in your animal physiology or anatomy, when you release it, what happened? This will be together connected with troponin, and troponin usually is tropomyosin will be presented to them. Usually troponin C, and when this happens, they will have actin myosin cross bridge forming. Is that right? That's why the muscle can do contraction. So, did you learn about these? So muscle, middle is M line. This is we call the A line. And then between these A line is called a sarcomere. Then in the below is H zone. Then we have a pseudo H zone, all those things. And you have this guy comes out, which is called a myosin. And you have here is called actin. And you go here, this is myosin. And the cross linked, that will have muscle contraction. Now we want a muscle contraction because the human body have to move in, but for the meat, we don't want that. It's giving you tough meat. So what we do, we have to break down it. So what we do, we add a calcium. Is that right? The calcium, the cowpan will be activated. Then surrounding disease, we have a nebulin, titin, all those proteins. So this is a little bit of anatomy there, but the key thing is that acetylcholine is the most important neurotransmitter to regulate or guide the muscle contraction. And the Clostridium botulism could prevent that, could stop it. So that's why you had a floppy baby. But all those things, the wound and the infant botulism, it is honestly, you can prevent it sometimes. It's basically the accident. You can understand about that. But more important in our everyday life, what we have is food botulism. Food botulism, sometimes it is hard. So why? We often see a very special food, and very common in West Virginia, canned food. So what are the canned foods looks like? So inside, you're going to have a pickles there. You have a cucumber there. You may have some slice of the tomatoes there. So you have like a pickle, you have a cucumber, you have a tomato. What this tells you? These are fresh produce. Where they come from? It comes from the soil. Then some people will say, can we wash it? You can wash it. You're using chlorine, you're using peroxyacetic acids or lemon citric acid, so citric acid, acetic acid to wash, you're not going to kill everything. They still have something on the surface, especially like these endospore-forming bacteria. They have a protection structure. So they're going to be there. And once you canning it, the pH is going to be lower. Then it's going to cooling the normal bacteria. And then those bacteria will be dominant. So will cause a problem. Therefore, in the food microbiology area, the canned food, we give them a category. The reason is these food products, water activity around 0.85, pH is around 4.6. So we call this is low acid food. And low acid food is very tricky. It is a high risk for Clostridium botulism. So what they do, they have some methods we can do. Number one, lots of the places, they are trying to let the pH below 4.0. This is what the West Virginia Department of Agriculture, they have the standards to do that. Less than 4.0, so what they do, we can add citric acid. Or sometimes we add acid concentrate to lower down the pH. But often the time when you only lower down the pH, it may not be helping you. So what they have to do is you have to use pressure cooker. Once you use a pressure cooker, you cook at 121 degrees Celsius with 15 psi. That might be more successful to curing the Clostridium botulism. This is kind of like a small autoclave we're using in the lab. But what happened is, for lots of the local producers, what they do is all those cucumbers or tomatoes, they maybe do a blanching, which means using boiled water to boil about like 5 or 8 minutes. Obviously, you're killing all the other bacteria, we call it the vegetative cells, but for endospore, they still can survive. So just want to let you know, the food's botulism is a tricky one. Now, how do we know the canned foods has been contaminated with botulism? Obviously, something you can see. First of all, you have off flavor, or even off smelling. That means Clostridium could be grown there. Second, you have a milky liquid. And once you see the liquid inside, the canned food looks like a milk. It's kind of like an extremely turbidity. That is a Clostridium going to grow there. What are the other things? You see the extracted liquid. The liquid has been coming out of the lid. That means it's contaminated. Or you have dented lid or the can. If you have a dented area, which means it could be caused gas, or the bacteria could be already being cultivation there dramatically. So those are the things we talk about the botulism. Following the botulism, we're going to talk about this guy. Is Clostridium tetanus. So I usually put these things together. Clostridium tetanus. First thing first, in the United States, we should not be worried too much about that. Because we have this guy. Remember? Clostridium tetanus is which one? Corynebacteria, diphtheria, Bacterial pertussis, that's acellular, that's tetanus. So if you have a diphtheria, tetanus, DTaP vaccine, you do that booster all the time, you should not have a problem. So Clostridium tetanus most of the time is for the people that are not following the vaccine schedule or like a drug abuser. Those are the people is a high risk. Clostridium tetanus obviously could also come from the wound. It's more obvious. Now, what they do, instead of you have a weak muscle or a no contract, this is a highly to extremely contract muscle. So therefore, what will happen? I'll have lockjaw comes out. Now why? The reason is, this is an enterotoxin. We call it a tetanus toxin. They are blocking another neurotransmitter, which is a combined gamma aminobutyric acid and glycine. These two combined together is muscle relaxing. If you block these two, of course, the muscle is always contracted. And you can see the picture looks like that. And finally what happened, the people cannot do breast. So that's what happened for tetanus. Now for tetanus, basically, if it's very severe, it could be, people could be dying. You can use some antimicrobials, antibiotics or antitoxins. But the effectiveness is questionable. Botulism, there are specific toxin for that. We have A, B, C, D, E, F, G toxin. And in the United States, ABE toxin is responsible for 60%. If you have a botulism happens, what they do, they're going to have to call the CDC. CDC in the South area will be calling Atlanta Airport. The airport will shipping anti-equine botulism toxin immediately. Equine is horse, you know about that. Anti-horse botulism toxin immediately. At the same time, you're using antibiotics. That could be relieve some of the symptoms. You still have the life. Clostridium tetanus sometimes is really tricky. That one, because eventually it will be causing, looks like that, the intercostal respiratory muscle will be interfered. So that's really, so you know, understand about it. It's always from soft to hard. It's always easy. When you become hard, come back to soft, it's not a reversible reaction from the biochemistry standpoint. So that one fully rely on is on the vaccination. We talk about these. This is a picture tells you the botulism and the tetanus. If you do not understand the anatomy, and this will explain for that. Okay, can you see it? You have the muscle. Acetylcholine signals muscle M to contract. Botulism blocks release of the acetylcholine, and in the other hand, tetanus blocks release of glycine. Well, glycine is one of the chemicals guided the muscle relaxing, and you don't have a relaxing, then of course it's just going to be contract. So this is a picture which is explains the botulism and the tetanus toxin in an easy way for you to understand. Okay, next one. Clostridium perfringens. This is a guy usually come from the food products also. but the symptoms is pretty obvious, and it is hard to do the treatment. Clostridium perfringens. Clostridium perfringens will cause a very typical symptom is looks like that. Gas, gangrene. This is also called mild necrosis. What is this terminology goes? Mild means muscle. Necrosis means death. So, muscle death, it is a Greek word. What will happen? Clostridium perfringens usually come from the food, is abusively storage, which means it's not be following the good temperature control. What we are talking about is this dangerous temperature zone. If your food extended time keep in this dangerous temperature zone, if it has been touched the soil or it's been contaminated with Clostridium perfringens, it will be multiplication and dominant. And what happened is, in case if sometimes you have a cut cell, this bacteria could be in colonization. Once the colonization happens, it will dig a hole and break down part of the muscle. The reason is not only for the toxin, this pathogen also will generate collagenase. Obviously, you see this will break down collagen. That is a major chemical for connective tissue of the muscle. So once you have a dig a hole of the muscle, it's released, they're going to grow. What are they going to generate? Is gas. Once more and more gas generated, they have a pressure. So every single piece of this muscle can be separated. Once they separated, what will happen? The low blood pressure. So eventually what happens is your muscle does not have enough blood suppliers for the nutrients. Of course it's going to be decayed, then become mild necrosis. If these things happen, what they do, there's only one thing they can do is using antibiotics, antitoxicants immediately. And for these micronecrosis area, you have to do the surgery to take them out. So pretty traumatic, and I would say pretty horrible when you look about that. So the temperature control is very important for the food. And we always say is keep cold food cold, keep hot food hot. So this is a very dangerous temperature zone, especially like I share you the story. You go hiking or you could go to see a football game the whole day, and then you do a low temperature, long time cooking. Sometimes you cook about 55 degrees Celsius, using so-called Swiss wide cooking, French methods. The beef makes very tender. The reason is the beef, we talk about the juiciness, flavor, and the tenderness. We want a tender beef. It's always good. That temperature is dangerous because it's killing all the normal bacteria and let the Clostridium perfringens or Clostridium species growing up. Or multiplication grow dramatically, then cause all the problems. Okay, so this is gangrene. Now, luckily, Clostridium perfringens, we can do a very easy determination. What happened is, on the blood agar, you will see double beta hemolytic zone. That's the reason it's easy to do the determination. First of all, it's anaerobic. So you're going to have to grow the blood agar. Then after the blood agar growing, you see the transparent zone and you see the double beta hemolytic zone surrounded the colonies and connected with each other. Then bingo, that is Clostridium perfringens, usually 80%. Then you have an idea what it is, of course, based on the patient's history. So what he did last night, he did like suicide cooking for those products. So that's Clostridium perfringens. Okay, Clostridium difficile. That's the last one. This guy is kind of ironic. Clostridium difficile is a tricky one. What happened is, so Clostridium difficile. Clostridium difficile usually happened in the hospital. So this is another nosocomial infection. That means hospital acquired. So we already talked about the first nosocomial infection is methicillin-resistant Staphylococcus aureus. And the Clostridium difficile is the second one, is another one. So what happened, what type of the people will have is intensively, or we call it extensively, using antibiotics. So let's say you have a 70-year-old grandma in the hospital, have pneumonia, and she is intensively using amoxicillin for about 7 to 10 days. Then what happened? He had a water diarrhea, bloody water diarrhea, and in the colon area, you will see all these type of pseudo-membrane colitis. This is the one, is a typical symptom for Clostridium difficile. Why? You are using antibiotics more than 7 to 10 days. Antibiotics is killing bacteria. Most of the bacteria in the intestinal or in the colon area, remember colon area have 10 to the power of 12. You basically killed lots of that. So Clostridium difficile started to colonization, start to grow. And then if you do a colonoscopic, you will see, it looks like the picture. Pseudomembrane colitis. Those are pus, which means a dead blood cell. And then what happened, you have a black water diarrhea. So this is what happens. Now, the problem here is how are we going to do the treatment? So remember, we are doing extensively antibiotic treatments. We're killing the normal bacteria. The idea is to put the normal bacteria back. So there are multiple ways we can do. The first thing what we do, using another antibiotics. So for example, if you are intensively using penicillin, which is a beta-lactam, you could be switched to using vancomycin. And we will explain to you the principle behind it. Why we can use vancomycin. That sounds very ironic. We already use antibiotics for 10 days. Then I have to switch, use another one. The second method was using usually in the mild symptoms, drinking lactic acid bacteria-based products. And even sometimes smoothie will help. The idea is the fermented products The lactic acid bacteria, as a beneficial bacteria, you put them back. These two usually for milder symptoms. How about severe one? You have to do fecal transplant. That sounds horrible, which means other people's poop gonna put in your body. That sounds really terrible. But the idea is you already have the damage to normal bacteria in your colon, in your gut area. We're gonna have to put them back. We have to do it immediately. So the fecal sample from the other person, from the healthy person, has to put them back. There is a database for that too. Same as a blood sample, but this is not that popular, just because it sounds terrible. That's the only reason. Clostridium difficile, usually we say it is a hospital acquired. But 15 years ago, all of a sudden, people do the isolation, find that there's a lot in the food supply system. And I remember the USDA by the time founded a several projects, which is multi-million-dollar, to do the preference and also the tracing of the Clostridium difficile in the food supply chain. By the time, there's a little bit of time period, it's a hard research topic. But later on, maybe people find that it's not that often happens, so the research has not become hot anymore. But just want to let you know, they do have a certain period of time, people thinking this hospital-acquired pathogen could be going to the food supply chain. So we call it an under-researched, possible emergent foodborne pathogen. Okay? Okay, we're going to go over this table real quick. This table, which is showed you briefly about all these microorganisms, your infection and source of the infection. We will talk detail later. Here, the key point, this table, most of them on the top, we'll talk about is food infection. Lots of them at the bottom is talk about food intoxication. So Salmonella typhimurium, this is the one. Now, by the way, this writing is wrong. The people who made up this book doesn't even know the naming. The typhimurium should be non-italic. Salmonella typhimurium, this is a pathogen, a high risk group for poultry meats and egg products. The second one is kind of a story behind it called the Salmonella typhi. This is a different pathogen. Salmonella typhi and the Salmonella typhimurium is a different story. This is a big story behind it. If you search Salmonella typhi on the website, you will find this woman called Melon. This is a very tricky story. It is back about 100 years ago or 80 years ago. The lady works in New York City. One day she had a fever and coughing, sneezing, all those symptoms. Then go to see a doctor. And the doctor said it might be Salmonella typhi. Because also have a fever, it's a typhi fever. So he did some treatments and then she recovered. There are 3 people surrounded her and died. And then she has to change the job. And to change the job, he got contaminated again, and then the people surrounded her died again. It's about back and forth a couple of times. Around like 13 people surrounded her has been died. Then by the time the NYPD, the New York City Police Department, have to do the custody for her. And later on, she has to be isolated in some places. And in 1960s, she passed away because of the other things. Because it's a so magic pathogen. So the research area has been doing, get his sample to do. Took many years, until almost 90 years later, in 2003, in his spleen sample, the Stanford University scientists got isolated the Salmonella typhi, and they find this is packaged by a macrophage, which is a self-defense system. So it's very magic. She had this Salmonella typhi in her body, carrying the rest of her whole life, and protected by a macrophage, even she died. So Salmonella typhi is very special. What we are talking about, Salmonella typhi, murine, Newport, all those things, when we talk about in our last section of the lectures, which means Salmonella non-typhi. So Salmonella, we usually talk about is Salmonella non-typhi. There are 6 subspecies. And the typhimurine is one of that. The full name should be Salmonella enterica subspecies enterica typhimurine. It's a long name. And the enterica is one of those 6 subgroups. So just want to let you know, Salmonella, it's not that easy. There's a lot of story behind it. Vibrio chloro. This is one. We had slides here we'll be showing you already. This is usually the seafood. It's a comma-shaped gram-negative bacteria. Shellfish is one of that. Any seafoods could contaminate it. Once it's contaminated, what will happen? You have a severe rice water diarrhea. What this means? You're going to lose tons of water in your body in like 1 hour. So the treatment for chlorella is not to using antibiotics. Is you need to do rehydration, which means put the water back immediately, plus antibiotic therapy. Now why? This is I said all the time. You look at here. Ideally, you should have ATP. This is an energy currency. Unfortunately, this enterotoxin, the chlorotoxin, instead of you have ATP, you have CAMP, cyclic adenine monophosphates. Once you have a cyclic adenine monophosphates, what will happen? The energy cannot be used, and it's locked there. So like the energy money is locked in your pocket. That's why it will cause a problem. And whooping cough is the same thing. Whooping cough in the respiratory tract cilia, the cilia area is also not going to have this happens. It will have this way, then the CAMP will be generated. This is a very basic mechanism why lots of the bacteria cause the disease. Instead of you have ATP, you have CAMP created. So that's for the chloro. Shigella. Shigella, it's a Shigella toxin. What happened for Shigella, most of the time it happens in daycare for the kids. So what happened at the daycare? The kids go to the restroom, then they just do not wash hands and touch here, touch there, then their poop sample is contaminated. So causes Shigella. Shigella most likely is in the daycare facility. Campylobacter jejuni, we'll talk about in our last lecture. It's another poultry risk pathogen, microaerophilic. E. coli, there is a lot. We'll talk later on. E. coli, this is one called travelers diarrhea, is one of the type of the E. coli. Yersinia. Yersinia usually not happen very often. It's happened in the pig chitlings. And you don't eat it, I eat sometimes. What this means? That's a pig intestinal. You don't eat it because in the United States, a pig intestinal is become a byproduct to making the casing of hot dog. But if you go to a Chinese restaurant in Pittsburgh, in Forbes Avenue, you can buy a bowl of pig chitlings about $15 or $20. It's spicy and really good. But if those raw sample of the chitlings are not sanitized very well, it's easily to contaminate with Yersinia. That is what Yersinia for. Of course, milk, these are, it depends, the pasteurized or new fresh milk or unpasteurized, it could be the vehicle for these pathogens, but it's not really the big one. the most likely the source of infection is from the pig chitlings. Listeria mouse pathogens, we're going to talk detail later on. That's a very tricky one. Vibrio, paro, hemorrho, Lyticus, this is a shellfish, the same as a vibrochloria. Clostridium perfringens difficile, we already talked about that. So I think we're going to end up today, and this we'll talk when we come back. When we come back, we will talk about sexual transmitted disease. We finish this section, then we're going to move to antibiotics.